Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the lipids, and with that, majority of the components of that macromolecular fraction we have completed, and now we are going with the next concept that is related to carbohydrates. In the case of carbohydrates, we need to study about monosaccharides, disaccharides and polysaccharides. But the point you need to know is in the NCRT, they gave only polysaccharides. They did not mention any information regarding monosaccharides and disaccharides. But the problem is, if we don't know the basic points related to this, unnecessarily we will face problem in the next coming chapters. In different other chapters, these names we will use. And not only that, when we study about polysaccharides means what? Many small units of sugar should be attached. If you don't know individual sugars, then how we will understand the concept? So that is the reason why, even though they have not mentioned in the NCRT, at least some basic points. Here also, please remember, since they have not mentioned and I am teaching it for easy understanding, I am also not exceeding the limit. Within the limit, whatever the least possible, only that much information I am trying to give you in the better time. Okay, please understand all these things. But another point I am telling you is, in this lecture we are mainly studying the structures. Please remember the structures. So my suggestion is, please better practice these structures so that it will become very easy when you are reading this NCRT content. And not only this, anyhow these structures once again you need to study in chemistry also. So that is the reason why I am saying that please practice all these structures. I will tell you how to interlink with all these things. Okay. Anyhow, regarding ribose, deoxyribose, already we studied the structures. So simply I will show you, and I don't want to spend more time on it. But for remaining others means mainly we will focus on glucose, galactose, mannose, and then fructose. These are the four things with structures we need to understand. The point is, if you know the structure of glucose, based on the structure of glucose, you can write all the things. How you can write all the things, that I will tell you. So that's why observe that content carefully, and later on you please practice it so that it will become very easy. Understood or not? So please try to practice on your own so that it will become very easy for us. And now we are going with the carbohydrates. Even though this basic, in this lecture whatever we are studying, it was not given in NCRT, so you cannot get any information for this. So listen very carefully here. So generally, what are these carbohydrates? Carbohydrates. Generally, initially when they have given this molecular formula, what they did, they made it as carbon hydrates. So that means they gave the molecular formula by keeping in view that they are the hydrates of carbon. This is the carbon. Hydrate means what? Water. So in this way they gave carbohydrates they gave. So that's why in the general formula we are generally talking like this: Cn, H2n, H2O n times. This n is the number of carbons. Based on this we are saying. But the point is, this formula is applicable only for some monosaccharides. This formula is applicable only for some monosaccharides, not for all carbohydrates. This is the another point that you need to remember. Even though we are saying that they are the hydrates of the carbon, originally they are not hydrates of carbon. So here there is no separate water molecule here. Please understand, as per the molecular formula you can write like this, but originally they are not hydrates of carbon. So please remember this. Even though they are called carbohydrates, they are not hydrates of carbon. This is the first point you need to remember. And the molecular formula we are generally using like this, and this formula is applicable only for some of the monosaccharides. That's it. Even not for all monosaccharides also. Majority of the monosaccharides will follow this formula. That's it. That is the better word. Okay. So in this way we are using this. That's why, what is the better definition for these carbohydrates? So carbohydrates are polyhydroxy. Carbohydrates are polyhydroxy aldehydes are ketones. They are polyhydroxy aldehydes are ketones. First we will see what is this meaning. Poly means many, hydroxy means hydroxyl groups. That means OH groups are present in many number. So many OH groups are present. And along with this, either aldehyde means what? CHO group, or ketone group is present. Observe this carefully. Anyhow, many hydroxyl groups are present. Along with this, either aldehyde or ketone. So between these two, only one will be there. Some are having aldehyde group and some are having the ketone group. Understood or not? In this way, they are present. Slowly we will go with the structures also. So you don't need to worry about this. Some important structures, not all, only some important things that I am discussing here. Okay. And in this way, we are talking about these carbohydrates. Before directly going into the structures, first we will see the classification. These carbohydrates are classified as monosaccharides, oligosaccharides, monosaccharides, oligosaccharides, and polysaccharides. Okay. Actually, what are these saccharides mean? Saccharides mean sugars. Saccharides are sugars. Okay. So monosaccharides. So single unit is present. That's why they are called as monosaccharides. In the case of oligosaccharides, generally 2 to 10 units are present. In the range of 2 to 10 units are present in the case of oligosaccharides. That's why oligosaccharides are again divided into disaccharides, trisaccharides, tetrasaccharides, pentasaccharides. Likewise, you can write 2 units disaccharide, 3 units are present trisaccharide, 4 units tetrasaccharide. Likewise, 2 to 10 units are present in the oligosaccharides. Poly means what? Many units are present. So many are present. In the polysaccharides again, different types are there. That separately under polysaccharides we will study. In this lecture, we will try to focus on this. We will try to focus on mono and oligosaccharides. In this one also, our main focus is monosaccharides and three important examples of disaccharides. That's it. We are not worried about tri, tetra, all these things. We don't need this. Only this basic information is enough, and in this lecture we will try to complete this information. So anyhow, regarding polysaccharides they gave in NCERT. In the next lecture we will go with. But this tri, tetra, they are not needed, so simply ignore that concept. Clear? And first we are going with monosaccharides. Only single unit is present. So that's why what we say that these cannot be further hydrolyzed. Why you are saying like this? Polysaccharides upon hydrolysis they will form oligosaccharides. Oligosaccharides upon hydrolysis they will form monosaccharides, and monosaccharides are the simplest units. They cannot be further hydrolyzed. That's why these are called as monosaccharides, single units, single sugars. Okay, single sugars. This is how we are saying. So these are the monosaccharides. And now we are going with classification of monosaccharides. And here we are classifying the monosaccharides based on the functional group. How we are classifying them? Aldoses. What are aldoses? Already we studied this word. So when we studied about ribose sugar, we studied that ribose sugar is a aldose sugar. What is the aldose sugar? Aldehyde group containing sugars are called as aldoses. And some are ketoses. Ketoses means ketone group containing sugars. Aldehyde group containing are called as aldoses. Ketone group containing are called as ketoses. This is how we are writing. And this is based on the functional group. And now at the same time we are also talking about the number of carbons. Based on number of carbons. If 3 carbons are present, it is called as a triose. If 3 carbons are present, it is called as a triose. Tri means what? 3 carbons. Next one. If 4 carbons are present, it is called as a tetrose sugar. Yes or no? If 5 carbons are present, it is called as pentose. One example I will give you. For each one example I will give you. Why I am giving these examples? Because these names mainly you will study under the photosynthesis chapter. You will study about the C3 cycle and all in the photosynthesis. There you will use these names. And not only there, in the respiration chapter you will study about glyceraldehyde, dihydroxyacetone, many of these. So these names whatever the names I am writing now, these names regularly we will use or frequently we will use in different chapters. So that's why at that time you should not get confused. So that's why here itself I am giving these basic names. Anyhow, some extra names are there, those you will study separately, but these are regularly used names, so every student should know. So that's why we are writing like this. 6 carbon containing is called as hexose, and the 7 carbon containing is called as heptose. Understood or not? So in this way we are writing these particular names, and now we will write the examples. One example I will write. 3 carbon containing, in that also aldehyde group containing, it is glyceraldehyde. In the glycolysis, in respiration chapter you will study this glyceraldehyde 3 phosphate, you will study, no, this is the glyceraldehyde. And then there itself you will study about dihydroxyacetone. This is also 3 carbon containing but ketone group containing sugar. 3 carbon containing ketone group containing sugar. Okay, that is one example. 4 carbon containing, here erythrose, 4 carbon aldehyde group containing sugar. Here it is erythrulose. You can easily understand the difference. Erythrulose, small difference in the spelling. 4 carbon ketone group containing sugar. Under the 5 carbon containing, we will talk about ribose sugar. Already we studied about the ribose. Corresponding to ribose, here the ketone group will be ribulose or ribulose. Anyhow you can pronounce this. You can pronounce this as ribulose or ribulose. Anyhow you can pronounce. And here not only ribose. When we studied about that DNA nucleotides and all, at that time we studied about 2-deoxyribose. This is also one important example. 2-deoxyribose. Okay, and these are the examples. Don't worry about this corresponding one. Under the hexoses, 3 important examples are there. Under the aldoses, glucose, galactose, mannose. These are the 3 important, very, not only this, so many are there, but these are most regularly used names. Glucose, galactose, mannose are 6 carbon aldehyde group containing. Under the ketone group containing, the most important example is the fructose. Under the 7 carbon containing, it is glucoheptose and galactoheptose. Likewise, different are there. And here it is pseudoheptulose. So in this way, different types of monosaccharides. So all these are monosaccharides only. Based on number of carbons and based on the type of major functional group, we have classified like this. Understood or not? I don't want to repeat this again. These are the various names. Frequently, very frequently, we will study about all these things. And out of this, now we will focus on ribose, deoxyribose. But anyhow, already we studied. Simply, directly I will show you the structures. And I will draw the structures for glucose, galactose, mannose, and then fructose. And one point for each one we will study. With that it will be completed. That's it. Clear? With this monosaccharides will be completed. So mainly we are focusing on the structures. So please observe these structures very carefully. Clear? So this is what. So in this way, carbohydrates are polyhydroxy, aldehydes are ketones. We are classifying them into monosaccharides, oligosaccharides, and polysaccharides. Under this, first we are talking about the monosaccharides, and this is what the table that we made just now. Understood or not? Now here, based on the functional groups, aldehyde group containing are called as aldoses, ketone group containing are called as ketoses, and 3 carbon containing trioses, 4 carbon containing are tetroses, 5 carbon containing are pentoses, 6 carbon containing are hexoses, and 7 carbon containing are heptoses. In this way, based on number of carbons and the type of functional group, different names we have given for these monosaccharides. Clear? With this basic information is completed. Now we will go with this. Monosaccharides. Majority. Please remember, not all. Majority of the monosaccharides, they will follow this particular formula. And here what is this N value? Number of carbons. Suppose it is 3 carbon. C3. H. 2 into 3 means 6. H6. O3. So C3H6O3. So in this way you can use it. Suppose it is for the glucose, C6H12, 2 into 6 is 12, right? So H12 O6. So it is following. So in this way majority of these are following this particular formula. Yes or no? So majority of these are following this particular formula. And if you clearly recollect it, when we studied about ribose structure, I told you that it is following. It is C5H10O5. It is in the formula of CnH2nOn. Yes or no? It is following the same thing. Yes or no? Next one. And here in the beginning itself I told you, no, they are the hydrates of the carbon. This particular formula you can write like this, or else you can even write like this. CnH2nOn, right? You can write like this, or you can write CnH2ON. Both of them are same only. H2nOn. Ultimately you will get the same thing. So you can write like this or you can write like this. But please remember, majority of the monosaccharides are following. Up to that it is okay. But who is not following this? Who is not following this? 2-deoxyribose is a monosaccharide, but it will not follow because its molecular formula is C5H10, but it is O4, it is not O5 because it is 2-deoxy, right? Even though it is not following the formula, still it is a monosaccharide. That's what I am saying. Some monosaccharides do not follow this particular formula, but still they are monosaccharides. Yes or no? In the same way, I will tell you one more example. If you are talking about the acetic acid, what is acetic acid? CH3COOH. You just write this acetic acid in this particular formula. How many carbons are there? 1, 2. 2 carbons are there. Hydrogens, how many are there? 3 plus 1, 4 are there. How many oxygens are there? 2 are there. So C2H4O2, it is following this formula. But this is acetic acid, it is not a sugar. This is not a sugar. It is not a sugar. Please observe carefully. In this way, even though we are using this generalized formula, some sugars are not following this formula, and some molecules, even though they are following this formula, they are not sugars. So in this way, exceptions are there on both the ways, That's why what we are saying, majority of the sugars are following this particular formula. This is the better word. Okay, so that's why we are saying like this. So in this way, these are the molecules. And now directly in the PowerPoint, I will show you the ribose structure and all, because already we studied about this particular one. Important pentoses. Under the pentoses, already we studied about the ribose sugar, ribose structure. The same ribose information we are writing in the furan ring. Furan ring, already we studied about this one. And then this is the 2-deoxyribose. The same information we are writing in the furan ring. So with respect to ribose, deoxyribose, already we studied the structures when we studied about the nucleotide components. Yes or no? So this part is completed. So now we are directly going with 6 carbon containing hexo, 6 carbon containing aldoses, aldehyde group containing. Under the 6 carbon aldehyde group containing, we are studying about glucose, mannose and galactose. Understood or not? So here what exactly we are studying here. So observe this content carefully here. So first we are writing about the glucose structure. First we will write the glucose structure and then we will write the mannose structure. Okay. So please observe carefully. So in the case of glucose, first at the middle I will write the glucose in the same way, whatever they have given in the same way. In the case of glucose, total 6 carbons are there. 1, 2, 3, 4, 5 and 6. Last carbon is CH2OH. And please remember, it is aldehyde group containing. So aldehyde always comes in the first position. This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Now you need to write OH and H. On which side OH is present, on which side H is present. Observe carefully. Here it is OH, here it is H, OH, OH. So now what happens? Here it is H, H, OH and then H. That's it. This is the glucose. This is the glucose structure. Clear? This is how the glucose structure is present. And now we are writing about the mannose structure. Mannose structure. For the mannose structure, here also 6 carbons, 1, 2, 3, 4, 5, 6. Last one is CH2OH, write as it is. First one is CHO, write as it is. Okay. And here if you are observing, once you know the glucose structure, where exactly you will get the difference here? So here we are getting difference at the second carbon. See here, here these two they are comparing, right? This is the Mono structure. At the second carbon only difference is there. Remaining structure is exactly same. So please observe this carefully. So at the second carbon what happens is, second carbon, here it is H, here it is OH. It is reversed here. But remaining is as it is. Remaining is as it is. That means here it is H, OH, OH, H, H and OH. I hope now you understood this. That means the difference we are observing only with respect to second carbon. That's why glucose and mannose are C2 epimers. C2 epimers. What is the C2 epimers means? Second carbon epimers. That means the position change we are observing only at the second carbon. Remaining structure is exactly same. So that's why glucose and mannose are called second carbon epimers. Clear? Now we will talk about the galactose. Here also we will compare with respect to the glucose. Now you can see it. In the case of glucose, what they are saying, at the fourth carbon there is a difference. Yes or no? Here OH on this side, H on this side, but here it is reversed. Only at the fourth carbon. Remaining all are exactly like a glucose. So here also, first let us write the 6 carbons. 1, 2, 3, 4, 5, 6. Last one is CH2OH, first one is CH. So first we will write this. And then here what we are saying, at the fourth carbon only there is a difference. Here on the fourth carbon what is there? Right side OH, left side H. So here what we need to write? Left side H, right side OH we need to write. And for remaining all, it is as it is like a glucose only. That means here it is OH, H, and here it is OH. Observe carefully. And here it is H, OH, and then H. Over. That's it. So here what happens between glucose and galactose, where we are observing the change? On the fourth carbon. So these are fourth carbon, C4 epimers. Fourth carbon epimers. This is how we are writing this. This is how we are writing these particular structures. Understood or not? And now what I will do is, now we need to represent this particular information in the form of Haworth's projection. When we are representing this information in Haworth's projection, first I will write for the glucose. 1, 2, 3, 4, 5, 6. CH2OH. This is the CHO, first carbon, second, third, fourth, fifth. This is OH, H, OH, OH. This is H, H, OH, and then H. Okay? This is what the structure of glucose. And the same information we need to write in pyron ring. Please remember, when we are writing the information of ribose sugar in that ring, we used a furon ring. What is the furon ring? Furon ring was like this. It was having 1, 2, 3, 4 carbon 5-member ring, and that is the furon. But this is the pyron ring. In the pyron ring, how many carbons are there? 1, 2, 3, 4, 5 are present. Then where we are mentioning the sixth one? Sixth one will be represented above. Understood or not? Sixth one is represented above. So in this way, within the ring, 5 carbons, but 6 members are present. Understood or not? So that's why from the fifth one, sixth one is projected away. This is how we are writing. So how you will write this one? Those which are present on the right side, they should be mentioned below the plane. Those which are present on the left side, they will be represented above the plane. Yes or no? So here what happens? So here also, so like ribose sugar, here also we are making ring structure between these two, last chiral carbon and the aldehyde group. So because of this, what happens? Because of this, there will be a rearrangement. So this will be like a circular form like this. And here you will get 2 options. What is the first option? OH on right side so that H is on the left side. This is one option. Second option is OH on the left side so that H is on the right side. This is how we are writing. So please remember, if you are writing like this, it is the beta form of the glucose. It is the beta form. And here OH is on the right side, capital D, glucose. This is how you are writing beta capital D glucose. Suppose you want to write alpha. In the case of alpha, what happens? Now OH is on right side, H is on the left side, this is on the right side. So that now what happens? Now this will be the alpha form. Already I told this particular information when we studied about ribose sugar. Yes or no? Ribose sugar. So the same content here also. This is how we are observing. Let us keep it as alpha form only. So alpha form means on this carbonyl carbon OH is present on the right side. So alpha form, alpha D glucose. This information let us write here. So how we will write this? Those which are present on the right side, they should be mentioned below the plane. First carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. Yes or no? For the first one, see, remember, anyhow for the fifth, sixth, we are not worried. Sixth is CH2OH, right as it is. For the fifth, already this O is connected to this first one. So already O connected. This hydrogen, you can simply write like this. So for this, you don't worry. We need to focus more on these first four carbons only. First four carbons only we should be careful. Those which are present on the right side, they should be written below the plane. So OH below the plane. For the second carbon OH below the plane. Third carbon hydrogen below the plane. Fourth carbon OH below the plane. This is how we need to write. And then those which are present on the left side, we should write above the plane. Now here hydrogen above the plane. Here also hydrogen above the plane. Here OH above the plane. Here hydrogen above the plane. This is how we need to write. And this is the pyranose form. So this is the alpha capital D pyranose, glucopyranose. Glucose, right? It is the glucose in the pyron ring form. So glucopyranose. Glucose in the pyron ring form. That's why it is glucopyranose. So in this way we are writing this. Understood or not? In the same way, for all these things also we can write. So please remember, for all these things, when we are making the ring forms, alpha forms, beta forms are possible. Alpha forms, beta forms are possible. If it is a linear one, on the right side, if OH is present, it is alpha form. In the ring form, what happens? On the carbonyl carbon, if the OH group is present here, suppose this is the plane. Below the plane OH is present, it is alpha. Suppose it is reverse, it will become reverse. That means above the plane will be the beta. So below the plane OH, it is alpha. Above the plane OH, it will become the beta. So according to the conditions, according to the structure, we can change this. In this way, this is what the structure with respect to the glucose. Same way you can write for the mannose, galactose, for all the structures. If you know for one, remaining also you can write easily. Understood or not? This is how we are writing this. Clear? This is how we are writing for this particular hexoaldoses. Now we are going for the hexoketose. There we are studying about the structure of fructose. Fructose. For the fructose structure also, first let us write the glucose again. If you know the glucose structure, you can easily write all other structures. 1, 2, 3, 4, 5, 6. CH2OH, first one is CHO, first carbon, second, third, fourth, fifth, sixth. This is OH, H, OH, OH. Please practice the structures. Whoever it is, we need to practice the structures or else we will make the mistakes. Okay. So this is what the structure of the glucose. Now fructose is also having the 6 carbons. So write the 6 carbons. Here I will write the carbon. 1, 2, 3, 4, 5, 6. CH2OH. But please remember, fructose is a ketone group containing. So ketone is never present in the first carbon. So here it is present on the second carbon. Ketone is present on the second carbon. So first carbon again it will become CH2OH. Yes or no? This is the first carbon, second, third, fourth, fifth. And now what happens? Now for 3, 4, 5, what you will write? Whatever we are writing for glucose, as it is, you just write H, OH, OH. So this is how we are writing. So that's what I am saying. If you know the structure for the glucose, it is very easy. You can easily write the all other structures. This is what the fructose structure is. And now this particular one, we need to represent in the form of a Haworth's projection. But please remember, even though 6 carbons are present, see, for the glucose, which ring we have used? Pyron ring we used. Pyron ring we used for the glucose, galactose, mannose. But for the fructose structure, generally we are using again furon ring. Again we are using the furon ring, like ribose sugar, here also we are using. But the problem here is, here only 4 carbons are present in this. So what are these 4 carbons? Observe carefully. This is the second carbon, third carbon, fourth carbon, fifth carbon. Then where you will represent 1 and 3, 1 and 6, 1 will be projected, 6 will be projected. So 6 will be projected here. This is the sixth one. Okay. Now 1 position, 1, how you will write the 1? As of now I am writing 1 here, but based on alpha and beta forms I will change the position of 1. Don't worry about this. This is how we are using the furan ring for the structure of this fructose. Then how we will write? First one CH2OH, write as it is. Okay. But for this ring structure, what we need to do, you need to convert this into ring form. So when you are converting this into ring form, last chiral carbon with carbonyl carbon. Observe carefully. Now these two will form the ring here. Understood or not? Now what happens? Now what happens? On this second carbon, this is the second carbon, right? This is the second carbon. On the second carbon, now how they are present? If first carbon is present like this, CH2OH, and OH is present like this, observed or not? That means OH is present on the left side. Left side means it will be beta form. It will be beta form. It will be like this. Suppose it is reverse. Suppose it is reverse means what? First carbon CH2OH on left side, and here OH is on the right side. OH is on the right side, then it will be beta form. Do you able to understand this? In this way, alpha and beta will be changed in this case. Clear? This is how we are changing. Now let us keep this as beta form only and let us write. Already CH2OH. How you will write this? This information should be below. So this OH is coming here. For the second, this is CH2OH is coming above. Suppose it is alpha form, what happens? You need to change this. So you need to change this. Now here what happens? OH will go above, CH2OH will come below. That's it. So that's why this will be beta form in this case. Understood? For remaining, for the third carbon, H below, OH above. For the fourth carbon, OH below, H above. For the fifth carbon, anyhow in the ring, so this hydrogen directly you can write it. This is how you are writing. So now this will be beta fructose. Okay, but this is beta fructo furanose, because in the furan ring we use it. This is the beta fructofuranose. So this is how we are writing this fructose structure. In this way, generally we are writing this glucose, fructose, galactose, mannose structures like this. With this, these structures are completed. Now we will see mostly one point for each of these particular structures. This is what the epimers concept. So glucose and mannose are second carbon epimers. Glucose and galactose are fourth carbon epimers. So please remember this particular concept. Okay. Already I wrote this particular information here. So next one, these are the ring forms and all. You please practice on your own. These things, when you are looking at the board, they will appear very easy. But when we are really practicing, not only for you, even for us also, it is difficult. If we are not practicing, we will make the mistake. So please practice. Okay. This is what. And now first we will see about the glucose. What are the major important points for the glucose? Glucose is also called as blood glucose, because in the blood, sugar, whatever we are saying, generally whatever the sugar that we are measuring in our blood, that sugar is actually glucose levels that we are measuring. So that's why glucose is also called as a blood sugar. Okay, that is the one point. Next one, glucose is the major form of the energy source. So when we are eating this glucose, please remember our body, see, generally we are having some preference for the carbon source, energy source. Suppose you are eating the normal sugar. What is the normal sugar we are using in our home? That is the sucrose. Suppose sucrose, glucose, both of them are taken. Now our body will quickly use the glucose first. So that's why preferred carbon source is the glucose for us. Please remember these words. In MBA, there is a lack of run concept. Once again, I will use these names. When two different energy sources are there, one is used first when compared with other one. The one that is used first is called as preferred carbon source, preferred energy source. So for the human, it is glucose. So that is the reason why whenever we are playing games, suppose we got weak, then what we will do? We are taking that glucose first. Instead of taking the normal sugar, we are taking glucose, because glucose is directly taken and it will be directly used for energy. Suppose you are taking the sugar, normal sugar called sucrose, it should be broken down first. and then it will release glucose and then fructose. Yes or no? Again that glucose will be used. So in this way it will take more time. Suppose you are directly eating glucose, directly it will be absorbed and it will be used for energy source. That's why glucose is the major form of the energy source. Whatever the food that we are eating, ultimately it should be converted into glucose so that it can be used as energy source. So please remember this one. And not only this, so glucose is also called as a grape sugar, because generally, please remember, fructose is called as a fruit sugar, because in the majority, please remember, in the majority of the fruits, fructose is the major sugar. Understood or not? But in the case of grapes, glucose is the major sugar. So that's why glucose is also called as a grape sugar. Please remember this. In this way, some points are there. Next one is regarding this fructose. Fructose is the ketohexose, 6 carbon containing ketone sugar. It is also called as a fruit sugar. It is called as fruit sugar, that is correct, no problem. But when some of the faculties, it is actually not the problem of the students, it is the problem of the faculties. When they are preparing the questions, what they are doing, fruits contain which of the following sugar? They are preparing question like this, fruits contain which of the following sugar? Glucose, fructose, sucrose, like this they are giving. Please remember the concept here. Don't think that fruits are having only fructose. Every fruit will have different types of sugars. But still, why fructose is called as fruit sugar? Because even though many types of sugars are present, major content is fructose. Sorry, major content is fructose. That is the reason why it is called as a fruit sugar. It does not mean that only fructose is there, glucose is absent. No, that type of questions if you are encountering anywhere, simply ignore those questions. Question itself is wrong. Please remember this. So in majority of the fruits, major form of the sugar is the fructose. That's why fructose is called as a fruit sugar. So along with the fructose, other forms like glucose, sucrose, they are also present, but their quantity is lesser when compared with the fructose. That is what the meaning is. Because I saw one question like, so fruits contain which of the following sugar? They have given the option glucose, fructose, sucrose and some other sugar they give. That is a blunder mistake. So fruits are also having different types of sugars also. I hope now you understood the concept of this. So even though fructose is called as a fruit sugar, but grape sugar will be the glucose. There is a separate point is there. So please observe carefully. Next one, this fructose is rich in, means more quantity, in the major quantity it is observed, seen in honey also. Here also there is a mistake here. So many faculties, they are preparing a question that honey contains fructose only. That is wrong. Not just fructose, glucose, sucrose, there also many other components are there. But here fructose, major sources of the fructose is honey. In that sense they are saying this particular sentence. So please understand this. Next one, it is also present in the seminal fluid. So whatever the semen we are saying, in that semen along with the sperm cells, even the fluid is also present, that is called seminal fluid. In that seminal fluid this fructose is present. And what is the role of fructose there? This fructose will be used by the sperm cells for their survival. Yes or no? For the sperm cells, see, until they are doing this fertilization, until then they have to survive. From where they are getting this energy? From this seminal fluid, they are getting this particular fructose and ultimately it will be used for energy production. So in this way, seminal fluid is also having the fructose. This is also another important point mentioned in human reproduction chapter of class 12th. So please remember this particular point. These are some important points related to the fructose. Clear? With this, monosaccharides are completed. And now we are talking about disaccharides. But please remember, I am not spending much time on this. Simple basic information I will try to tell you here. Clear? And here we are studying about 3 major important examples of disaccharides. Disaccharides will have 2 units. What are these 2 units? In different disaccharides, different types of units may be present. First important example we are studying is the sucrose. Sucrose, please practice the structures, then only you can understand this. So what are these structures? One is glucose, other one is the fructose. Observe this or not. This is the glucose, this is the fructose structure that they have given. And here one glucose unit is attached to one fructose unit. Very good. And this particular complete structure is called as a sucrose. And this sucrose is the table sugar. It is also called as a table sugar because normally in our home, whatever the sugar that we are using, that sugar is nothing but sucrose. So this one sucrose molecule has one glucose and one fructose in it. But if you observe this, glucose is in alpha form and fructose is in beta form. This is one important point you need to notice. Second point you need to notice is the bond. What is the bond that is present here? Between two sugars, it is called as a glycosidic bond. Please remember this. Please remember this name. Bond name is also very important. Glycosidic bond. Glycosidic bond. Please observe carefully. In the case of proteins we studied amide or peptide bond. In the case of DNA and RNA means nucleic acids we studied phosphodiester bond. And in the case of disaccharides, trisaccharides or simply oligosaccharides and even polysaccharides. In oligo and polysaccharides, what is the bond that is same? Glycosidic bond. Glyco means sugar. Sugars are involved in the bonding. That's why it is called as glycosidic bond. Please observe carefully. Glycosidic bond. Simply it is called as glycosidic bond. So please observe carefully. Here there is a glycosidic bond. And my suggestion is please observe what are the carbons involved in the bonding. In the glucose, first carbon involved in the bonding. In the fructose, this is the second carbon. The structure they are writing in the reverse. So please observe carefully. This is second carbon. First carbon of glucose, second carbon of fructose. Both of them are involved in the bonding. Its significance, I will tell you after completion of this. I will talk about reducing and non-reducing sugar. At that time I will tell you. So please remember. In the sucrose, first carbon of glucose, second carbon of fructose, both of them are linked. That's why it is alpha 1 of glucose, beta 2 of fructose. These two are involved in the bonding. So alpha 1 beta 2 glycosidic bond. So it is called as sucrose. Sucrose is called as a table sugar or the normal sugar. It is also called as the cane sugar because from this sugar is obtained from the sugar canes. That's why it is also called as the cane sugar. Understood? And it is also called as invert sugar. Please remember this word. Very, very important because even though digestion and absorption chapter is deleted, I will tell you about some enzymes, names and functions. You need to remember those. There, instead of using the word sucrase enzyme, we will use the word called invertase enzyme. Sucrase enzyme is also called invertase enzyme. What it will do? It will break the sucrose into glucose and fructose. Sucrase or invertase enzyme, what it will do? It will break the sucrose or invert sugar. Sucrose is also called as invert sugar into glucose and fructose. Now you can understand why it is called as invert sugar. It is based on the optical rotation. The rotation of sucrose is different and after breakdown whatever the hydrolysis product we are getting and their optical rotation is opposite. So since original sucrose and after hydrolysis, so these two are having opposite directions of plane of rotation of the rotation of plane polarized light. So that's why it is called as invert sugar. Inversion we are observing, opposite rotation we are observing. That's why the sugar is called invert sugar. So that's why sucrose only, sucrose, not these two. Only sucrose is called as invert sugar. Anyhow, this rotation of plane polarized light, all these things I don't want to teach in this chapter. You will study about these in chemistry biomolecules. Please observe this carefully. In chemistry biomolecules, you will study about this. This is called as a muta rotation, M-U-T-A, muta rotation. I don't want to teach about muta rotation. You will study this in chemistry and that day once again you will study about this. So please remember this. This is about the sucrose. Over. Next one is regarding lactose. In the case of lactose, what happens? Beta galactose and it may be either alpha glucose or beta glucose. If it is beta glucose, it is called a beta lactose. If it is alpha glucose, it is called as alpha lactose. So any molecule you can use, no problem. But this galactose should be beta only. Please remember here, this is beta galactose. Beta galactose. In the beta galactose, first carbon is linked with fourth carbon of either alpha glucose or beta glucose, whatever it is. This is galactose, this is glucose. First carbon of galactose linked with fourth carbon of glucose. This is how it is present. It is called as lactose. Lactose is also called as milk sugar. Lactose is milk sugar because in the milk, whatever the milk that we are drinking, in that milk also a form of sugar is present. The major form of milk sugar is lactose. Lactose is called as milk sugar. So please observe carefully. So now this lactose, what is the enzyme acting on it? Lactase enzyme. Suppose we are drinking this milk. Now in this milk, this lactose sugar is present. In our digestive system, it should be broken down. So who will break this? Lactase enzyme. So lactase enzyme will break this bond, so that galactose and glucose will be separated. So please remember this. And regarding this lactose, you also need to remember this, because in class 12th chapter called MBA, molecular base of inheritance, there is a concept called as lac operon, lactose operon. There we will study some enzymes, and that enzyme name is beta galactosidase. That enzyme also can break this particular bond. You need to remember this. In that chapter I will talk more about this particular sugar. Okay, this is about the lactose. And finally we are talking about the maltose. Maltose is also called as malt sugar. Malt sugar. What is this malt sugar? So generally in the germinating seeds, what happens? Seeds are storing the carbohydrates like starch and all. When starch is undergoing hydrolysis, if two units, if they are separated, that is nothing but the maltose. That's why it is called as the malt sugar. Please remember, this is called as malt sugar. In this maltose, what happens? Both the units are glucose only. Both the units are glucose only. This is alpha glucose, this is also alpha glucose. So first carbon of this glucose is linked with fourth carbon of this glucose. So in this way, here also 1 and 4 are involved. But in this case, first carbon of glucose, second carbon of fructose, both of them are involved in the bonding. So in this way, disaccharides, these are the few important, very important disaccharides that you need to know. In this way, sucrose has glucose and fructose, lactose has galactose and glucose, maltose has 2 units of glucose. In this way they are linked. Wherever it is, the bond is called as glycosidic bond. That's why we will say specifically alpha 1 beta 2 glycosidic bond. Here it is beta 1. Suppose this is also beta 1, beta 1 beta 4. Suppose this is alpha, beta 1 alpha 4, whatever it is, beta 1 to 4 glycosidic linkage. This is also, this is alpha 1 to 4 glycosidic linkage, alpha and alpha. First carbon of this, fourth carbon of that. So alpha 1 4 glycosidic linkage. In this way we are writing these particular bonds. With this, disaccharides concept is completed. And now we are talking about a concept called as reducing and non-reducing sugar. Reducing, non-reducing sugar concept was not given in NCERT, but reducing and non-reducing end concept they gave in NCERT. To understand this, you need to understand this. So now there is a simple logic. How you will decide that whether it is a reducing sugar or non-reducing sugar? So in the chemistry you will study some agents, some chemicals you will study. So what we will do is, when this sugar is added to that particular chemical, whether it is reducing that particular chemical or not, whether this sugar can reduce it or not. So how you will decide it? Simple logic. Already we studied about these carbohydrates. They are having either aldehyde group or ketone group. Anyone is there? Either aldehyde or ketone group is there. Now what you need to observe? If aldehyde group is free or ketone group is free, these free aldehyde or carbonyl groups, they can reduce that agent. So what you need to observe in this disaccharide structure, if anyhow each one is having one aldehyde or one its own aldehyde or ketone group, right? Please remember, between these two units, if at least one unit, if at least one functional, one carbonyl group is free, it can reduce it. Now you just observe. In the case of maltose, first carbon aldehyde is in bonding with fourth carbon of the next glucose. That means here aldehyde is not free. But for this one, first carbon aldehyde is free. Here it is the ring form, that's why we are writing like this. But if you are writing the linear form, now this is the carbonyl carbon, that is free. So at least this carbonyl carbon is free. So this can reduce that reducing agent. Okay. Failing solution or Bendick solution, likewise different solutions are there. In there some agents will be there. So in there some ions will be there, and those ions will be reduced. So if this sugar has the ability to reduce that, now it is called reducing sugar. So maltose is reducing sugar or not? Yes, it is reducing, because for this sugar carbonyl carbon is free, so it is reducing sugar. Same concept here also. In the case of lactose also, here carbonyl group is in bonding, but this carbonyl group is free. Since this carbonyl group is free, it is reducing sugar. So lactose and maltose are reducing sugars because they have free carbonyl carbon. But in this case, for the glucose carbonyl carbon is first carbon. For fructose carbonyl carbon is second carbon. These two are in the bonding. That means in the case of sucrose, neither in the case of glucose nor in the case of fructose, free carbonyl group is not present. In this structure, free carbonyl group is not present because both the carbonyl carbons are in the bonding. So since free carbonyl group is not present, sucrose is not reducing sugar. So that's why sucrose is very important example for examination. Sucrose is a non-reducing sugar. Sucrose is a non-reducing sugar, whereas lactose and maltose are reducing sugars. Even though this content was not given in this particular chapter of biology, but these are very important in the chemistry. Please remember this. In this way, this is one important exception, so please remember. Then what about the monosaccharide? Any monosaccharide you just take, they are individual units. Individual unit means aldehyde group or ketone groups are free. So all the monosaccharides are reducing sugars. Please remember this. All the monosaccharides are reducing sugars. In the case of disaccharides, lactose and maltose are reducing sugars, but sucrose is non-reducing sugar. Very important example, so please remember this. The same thing I have written here. And here in the next lecture you will understand this particular sentence, because in the next lecture we will study about polysaccharides. In the polysaccharides, many units are attached to each other. At one end, free carbonyl carbon is present. That's why that end is called as reducing end. On one end, carbonyl carbon is not free, so that's why this end is called as non-reducing end. But anyhow, regarding this particular one, once again under the polysaccharides I will explain you, so don't worry about this. So now you just understand what is reducing sugar and what is non-reducing sugar. A sugar that is having a free carbonyl group, maybe aldehyde, maybe ketone, whatever it is. If the group is free, that is called reducing sugar. If these groups are not free, that is called non-reducing sugar. That's it. With this, monosaccharides, disaccharides information is completed. Even though this was not mentioned, at least some very important basic concepts I have explained. At least this information please try to remember, because they may ask questions even in the NEET examination also. It is not optional, it is compulsory. Every student, at least this information you should remember. Okay, extra information is your headache, but at least this basic information you need to remember. Please this much is important. Okay, so with this lecture is completed. So in the next lecture we can go with the polysaccharides. Okay, we will meet in the next lecture. Thank you so much.